Sensory perception begins at specialized receptors that detect physical or chemical stimuli and convert them into electrical signals. Receptors are classified by modality, stimulus origin, and receptor distribution. Modality groups include mechanoreceptors, touch, pressure, vibration, thermoreceptors, temperature, nociceptors, pain, photoreceptors, light, chemoreceptors, taste, smell, blood gas levels, and proprioceptors, spindle fibers, Golgi tendon organs, joint receptors. Each receptor type exhibits a unique transduction mechanism tuned to its stimulus. Receptors also differ by location. Exteroceptors lie near body surfaces and monitor external stimuli. Interoceptors reside within viscera and blood vessels, sensing internal conditions. Proprioceptors are located in muscles, tendons, and joints, informing the central nervous system about body position and movement. At the receptor membrane, stimulus energy opens or modulates ion channels, producing a receptor, generator, potential. In non-spiking receptors, this graded potential directly triggers neurotransmitter release onto afferent fibers. In spiking receptors, the receptor potential modulates action potential frequency in the first-order neuron. First-order sensory neurons carry impulses from peripheral receptors into the spinal cord or brainstem. Cell bodies reside in dorsal root ganglia for body sensations and in cranial nerve ganglia for head and face. Central processes of first-order neurons enter the CNS and synapse in specific nuclei. Ascending pathways convey different modalities. Fine touch, vibration, and proprioception ascend ipsilaterally in the dorsal column medial lemniscus system. First-order fibers ascend in the gracile and cuneate fasciculi, synapse in the medullary nuclei, cross in the internal arcuate fibers, and join the medial lemniscus to reach the ventral posterolateral nucleus of the thalamus. Pain, temperature, and crude touch travel in the anterolateral, spinothalamic, system. First-order fibers enter the dorsal horn, synapse in the substantia gelatinosa or nucleus proprius. Second-order neurons cross in the anterior commissure, and ascend contralaterally in the spinothalamic tract to the ventral posterolateral thalamus. Face sensations follow analogous routes in the trigeminothalamic pathways. First-order fibers of CNV, 7, 9, and X synapse in the trigeminal sensory complex. Second-order neurons cross and ascend in the ventral trigeminothalamic tract to the ventral posteromedial thalamus. Other ascending tracts include spinoreticular, arousal and pain modulation, spinomesencephalic, orienting to pain, and spinocerebellar, proprioceptive feedback for coordination. In the thalamus, third-order neurons relay information to the cerebral cortex. The ventral posterior nuclei project to primary somatosensory cortex, area 3,1,2, in the postcentral gyrus, arranged somatotopically in the sensory homunculus. Each body region has a disproportionate cortical representation based on receptor density and importance. Primary somatosensory cortex processes basic attributes: location, intensity, modality. Secondary somatosensory cortex and association areas, posterior parietal cortex, integrate inputs for stereognosis, texture discrimination, and spatial orientation. Lateral inhibition enhances contrast and sharpens spatial acuity by suppressing neighboring pathways. Parallel processing streams carry specialized information. The dorsal stream links somatosensory inputs to motor planning for object manipulation. The ventral stream integrates touch with visual inputs for object recognition and perception. Descending modulatory systems regulate sensory flow. Corticothalamic and corticospinal projections adjust thalamic relay and dorsal horn excitability. The periaqueductal gray and rostroventral medulla form the descending antenociceptive system, inhibiting nociceptive transmission under varying behavioral states. Functional organization extends beyond static pathways. Experience-dependent plasticity refines cortical maps. After injury or repetitive use, cortical areas can expand or reorganize, underlying learning, rehabilitation, and phantom sensations. Throughout development and adulthood, the sensory system operates as an interconnected network: receptors, afferent pathways, thalamic relays, cortical processing, and modulatory feedback, all working in concert to convert physical and chemical stimuli into perception, guide behavior, and maintain homeostasis.